What I'm going to do today is to talk about the two items. First is the genetic tours, and then the second is going to talk about some of our new findings on the new variants of the SARS-CoV-2. So when we entered to work on SARS-CoV-2, we developed the reverse genetic system. So following the previous coronavirus reverse genetic platform, we developed a 7-fragment RT-PCR clone plasmid. And then by ligating those 7 cDNA that covers the whole genome, and then through T7 transcription and followed by transfection of the RNA representing the whole genome length into the viral E6 cells, we were able to recover very robust viruses that recapitulates the phenotype of the clinical isolate. We use the Washington State index case isolate. So very quickly, once we developed that, we thought it would be very useful to develop some reporter viruses that will be very useful for different research and countermeasurement development. The first thing we did is we put the mNeonGreen reporter gene into the open reading frame 7 of the viral genome so that we can create very robust reporter viruses. And using these reporter viruses, that allows us to do high throughput drug screening, as well as measuring the neutralizing titers against the authentic viruses. The platform is very high throughput, and you do it in the 96-well format or 384-well format. And in terms of measuring the neutralizing antibody titers, you see the cells on day 0, and next day you mix your viruses with the antiviral compound or serum, and then you load the mixture onto the preceded cells. 16 hours later, you just use the high content imaging to read the inhibition of the antibody levels against the green fluorescent cells. So using these, we partnered with Pfizer, and we performed all the preclinical and the phase 1, phase 2 neutralizing antibody levels for the Pfizer's vaccine. And we're very happy that the first one is the first COVID vaccine got approved in the United States. And then of course, this tool is very useful, but at the same time, it has a limitation. The major limitation is the requirement of the BSL-3. So then that is the question we want to address. Is there a way that we can culture the virus in the authentic way, allow the infection and the replication, but it can be done at the BSL-2? So this is the recent publication we had in the Cell. So we started to work on the transcomplementation system. We put a lot of effort before on the replicon system, and we didn't get any luck. So as an alternative, we started to explore the transcomplementing system. The concept is very, very simple, like many other viruses have been successfully done before. So what we did is in panel A shows the cartoon. What we did is based on the previous coronavirus research, we deleted 2 genes from the viral genome using the reverse genetic system. We deleted open reading frame 3 and the envelope open reading frame. And then also as a reporter, we retained the mNeon gene at the open reading frame 7. So if you transfect these RNA with these 2 genes deleted into the cell that can transcomplement the 2 missing proteins, that is inducible in this cell line, which is the viral E7 cell line, and we will be able to package and produce viral particles. And then these particles will, when they infect the normal viral cells or other normal cells, it will allow only the single run. It infects and replicates, and because of the missing of the 2 deleted genes, it's no longer to assemble to go for the next run. So that's the concept. To build in the safeguard into this system, one of the approaches we took is we mutated these TRS transcriptional regulatory sequences that is required at the beginning of the genome and at the 5' end of the subgenomic RNAs and mutated to a non-virus sequence. And this approach has previously shown that this will prevent the accidental recombination and creating the replicating viruses if there is some other viruses, coronavirus or wild-type viruses in the culture. And that's the safeguard we built into this. So this slide shows the first initial transfection of the transcomplementing system. So panel A shows you the 2-gene deleted viral genome transfected into the transcomplement cells. And then from 24 hours to 72 hours, we'll harvest the supernatant of the transfected cells and measure the TCID50 using the mNeon reporter positive cells as the readout. You can see it increases as time goes on, but the titer is not that great, 10 to the 4th TCID50 per ml. And then under the electron microscope, you can see the consistently particles. And then in panel B, I'm showing you the single round infection of the product after this electroporation transfection. So in the first round of infection, we're infecting the viral E6 cells. You can see 24, 48 hours, a lot of green positive cells showed up. If you take the supernatant from the first round of the infection to infect the next round of naive viral cells, you don't see any green positive cells, indicating that there is a limitation of the infection. It's a single round of infection. As I showed in the previous slide, that the initial electroporation or production efficiency of the single round particles is not very high, 10 to the 4th TCID50 per ml. So we would like to improve the yield of this system. So we started to continuously passage these particles. Instead of going to infect the naive viral cells, we continuously go back to the transcomplement cells and to enrich adaptive mutations so that we hope that we can get some more efficient packaging efficiency and improves the yield of the system. So this is what we did. And this is the brown bar. This is 24, 48 hours. The initial product, you can see, 10 to the 5th TCID50 on 48 hours. And after 10 rounds of passaging in the green triangle, you can see a significant increase of the yield. And when we sequence these P10 passage particles of the total genome, we found that the deletion is still there. The mNeon reporter gene is still there. And in addition, we found there are 3 adaptive mutations indicated in panel D. These are located in NSP1, NSP4, and the spike P812R. So then we started to engineer these adaptive mutations into our system to see what is required for the improvement of the efficiency. It turned out to be that all 3 mutations is needed to have a robust packaging efficiency from the system as indicated in black diamond. So these results demonstrate that, once we have adaptive mutations, the system can go to around 10 to the 6 TCID50 per ml. So then, of course, it's very important to look at the virulence, potential virulence of the systems in animal models. So after consulting with our IBC committee, we followed their advices and to test the safety of the system into 2 animal models. The first is the hamster model. So what we did is the intranasal infection of the hamster with the wild-type virus, and then also all using our transcomplemented particles. And then for all the animal work, we intranasally or infected animals at the highest possible dose, just trying to get to the bottom of the liability of the safety of this system in the animal model. So in panel B, you can see the weight loss. The Y-type virus infected hamsters had showed weight loss. And then for the transcomplementing system particles, even it's 6 times more, as I said again, this is the highest possible dose we can do with the viral yield we can produce, and there is no weight loss. And then in terms of disease, the different days post-infection, and you can see that the Y-type virus infected animals showed disease from day 5 to day 7. And throughout the course of the infection, the transcomplemented animals, there is no disease at all. And then we also using the RT-PCR to look at the viral load in the nasal wash, oral swab, and trachea and the lung. As you can see that the transcomplementing the RNA level is a couple of logs lower compared with the Y-type infected animals. And then through collaboration with Mike Diamond, we tested the transcomplementing system in a more susceptible animal model, which is the K18 human S2 mouse model. So we tested them using 2 different infection routes. One is the intranasal, and another is to directly inject them into the brain intracranial. So on the top panel, H and I, I'm showing you the weight loss. You can see the Y-type, 2,500 TCID50 intranasal infection, there is significant weight loss. And in the eye, panel I, there is a death. And then for the transcomplemented system, even there is a couple of logs more inoculum, and there is no weight loss and 100% survival. In the intranasal intracranial infected animals, we can again show that the Y-type virus infected animals, and they lost weight and they died. And then in contrast, the transcomplemented particle infected animals with the highest dose in this case, 60,000 TCID50, and there is no death at all. And for survival, even with the Y-type 5 inoculum with 5 TCID50, there is still death. So these data demonstrate that at least in hamster and the mouse K18 human S2 mouse model, these transcomplement particles, there is no virulence, there is no dissemination. So then we tested the usage of this system for neutralizing antibody activities using the convalescent COVID patient specimens. And for each of those specimens, we performed the plaque reduction assay, as well as the transcomplementing using the mNeon reporter as the reader. They correlate very well. And then you can use this for antibody activity testing and the small molecule, in this case, remdesivir, for the antiviral activity testing. So now I'm going to move to the new variant. So previous speakers already mentioned that the D614G in the spike might be the first predominant variant mutations appear in SARS-CoV-2. So we performed some studies trying to look at whether this D614 increases the fitnesses in the hamster model, as well as whether it increased the transmission, or what's its effect on the vaccine efficacy, particularly neutralizing activity of the variants. So using the reverse genetic system, we generate a pair of the isogenic viruses, and everything identical using the Washington index isolate, the strain, and then the D614, the original one, and the G614, the variant one. So the first experiment is we compare the growth kinetics on the CALO3 cells, which is the lung epithelium cells. So you can see on panel C, there is slight advantage for the G614 viruses in terms of replication kinetics. And in contrast, in panel D is the same specimen, the supernatant from the culture medium. We measure the RNA, the genome copies, and you can see the mutant viruses, the G virus, actually the RNA copy levels is lower. If you calculate the RNA PFV ratio, there is a slight increase that the RNA PFU ratio for the original virus is higher than the G614 virus, indicating the G614 might increase the infectivity of the virus. And then we move to the hamster model to look at the fitnesses in the respiratory tract. So we intranasally infect equal amount of the original D virus or G virus, and then we observe the weight loss, and they equally lost weight to a similar levels for indicating there is no disease severity differences between the wild type and the mutant viruses. And when we measure the viral load in the respiratory tract on day 2 in panel B, and then day 4 in panel C, we can clearly see there is some advantage of the G viruses indicated in red, and particularly in day 4, in the nasal wash and the trachea, there is clearly advantages, while in contrast, in the different lobes of the lung on day 4, the mutation did not seem to affect the viral load in the lower respiratory tract. So these data clearly indicates that the mutation has conferred advantages for the viruses to particularly replicate in the upper respiratory tract, indicating that might increase the transmission. To further demonstrate its human relevance, we performed the growth kinetics of the isogenic pair of the viruses on the human airway culture, primary culture. So in panel B, this is just to look at the growth kinetics of the original virus versus the mutant viruses. You can clearly see in the human airway culture, the mutant virus has good advantage. And then the RNA level, they're quite similar in panel C. And again, calculate the RNA PFU ratio indicates that the mutant virus increases the specific infectivity of the viruses. And then to further reconfirm those results, we did the competition experiments. So what we did is we mix in panel E, we mix 1-to-1 ratio of the original virus versus the mutant virus, the same amount inoculum to the culture. And then over the different days here in panel E, we calculate, sequence them, and look at the population ratio between the wild type and the mutant. As you can see, from day 1 to day 5, the mutant virus clearly wins the competition. And in panel F, we started to change the ratio of the input. So the wild type versus the mutant 3-to-1 ratio. Again, you see the mutant virus wins in the culture. Even if you, in panel G, I'm showing you 9-to-1 input ratio, and the mutant virus still able to outcompete the wild type virus. These data clearly indicates its advantage in the human airway culture. So then we also looked at the stability of the thermal stability of the viruses between the wild type and the mutant. And we incubated equal amount of infectious viruses at different temperatures, 42, 37, and 33, which mimics the upper airway temperature. Over the time, we measure the infectivity of the remaining PFU using plaque assay. You can clearly see that a consistent pattern that the mutant virus is more thermostable compared with the original virus. This might also explain contributing to why it is replicating to a higher level, has its fitnesses. And later on, both Yoshi Kawaka and the Rolf Barrick's group using the hamster model, they demonstrate directly transmission that the D614G enhances the direct transmission in the hamster. And also Dave Wentworth's Volkov and the Martin Beer's group using both hamster and ferrets model demonstrate that the same mutation really confirms the advantage in transmission among the animals. So these are the data on the D614. And now I'm going to switch to some unpublished data that is about the UK strain to look at the infectivity and the transmission of this new variant. And then even we're more interested if there is a determinant in those mutations that is mainly that is responsible for the enhanced transmission observed in the epidemic and the clinical setting. So what we did here is, again, using the reverse genetic system, we individually engineer the mutations, individual mutations. There are all deletions. There are 8 of them from the B117 variant and into the backbone of the Washington isolate. And once we recover all these mutant viruses, and then we use the competition experiment in the hamster model to look at the advantages of each of these mutations. So we use the competition, we use the Washington state isolate, bearing these D614G I just explained in the previous slides to compare with this, because the B117, they all have these D614G mutation already. So the experiment setting is very straightforward. We intranasally infect the hamsters, and on day 1 post-infection, those hamsters were infected 1-to-1 ratio, the each individual mutant, and then the Washington strain with the D614 1-to-1 ratio. And then we co-cage the infected animals with the clean animal for 8 hours. And then we separate them. And then we started to measure the viral loadings between the 2 viruses we inoculated. And then we calculate the ratio between the 2 viruses at each data point. So this is a day 4 from the donor animals. And then on panel C is the recipient animals to look at the nasal wash and the 2 populations for each individual mutant. So I know it's a busy slides, and we clearly see that N501Y mutation that consistently shows the advantage. And the previous speakers mentioned about the 6970 deletion. It also shows the advantages in the donor in terms of the nasal wash here. And then in the recipient, also you can see the 6970 deletion, and to a large extent that N501 mutation has an advantage. And then we also included a UK that contains all 8 mutations or deletions in the spike gene in the background of the Washington isolate. And then basically the 501 itself can pretty much mimic the complete spike mutation viruses. And then, so these data that point us to 501 is the major one that can recapitulate.
Pei-Yong, it's 2 minutes warning.
Sure. And then we focus on the 501, and we did, more detailed characterization from the donor and the recipient. You can see the donor from day 2 to day 3 to day 4, it has advantages, the recipient the same, and then the trachea and lung, they show the same pattern. And then in the human airway culture, it's the same. We did the competition, and for 501, it outcompetes with the original virus with the D614G. In terms of the mechanism, we did the human S2 and the RBD binding interactions. We clearly show that once you have the 501, the binding at the KD and the K off is improved by greater than 100-fold. So that really explains the mechanism by increasing the attachment and entry. So finally, through collaboration with Pfizer, we looked at the new variant spike, its effect on the neutralizing activity of the serum from the people vaccinated with Pfizer. So what we did, again, using the reverse genetic system, using the Washington isolate, we made the chimeric virus, which in the first one, the B117, we have the whole spike from B117, and for South African strain, the whole spike from South African strain variant, and the Brazil, same, the full spike. And then using the 20 vaccinated serum, we looked at the head-to-head, did the plaque assay reduction in the conventional way. And then this is the NT50 titers indicated. You can see the UK and the Brazil, the chimeric viruses, there is not very much, there is no significant differences in terms of the neutralizing activity. But in contrast, the South African strain, the neutralizing titer reduced by 3-fold. So among the 3 variants, as we all know from different groups, other groups study indicating that the South African strain is of the major concern. So I think I would just stop here and thank all the collaborators and people from the lab, particularly collaboration with Pfizer. It's been a great journey. And then the great colleagues at UTMB, Scott Weaver, Vineet Manisheri, and then also I mentioned Mike Diamond, collaborating on the animal work and the funding agencies. And I will stop here to answer any questions. Thank you.
Thank you, Pei-Yong. It looks like we have some questions from the audience. So Luis Kuhn, you should be able to talk if you unmute yourself. Are you there, Luis? If not, Wayne Hendrickson has his hand up. Wayne, if you unmute yourself, you should be able to talk.
But I don't have a question.
Oh, okay. Well, let's try number 3. Michael DeWild. You should be able to talk. Unmute yourself.
Yes, obvious question. Do you have any data on 484 alone?
484 alone, yes, I didn't include this, and those data I think we published in Nature Medicine maybe last month or so, and those data are there. Yes, for the South African strain, we even did some of the subdomains. For example, we mutated all the 3 RBD mutations we engineered into the Washington isolate. And it also has a reduction in terms of neutralizing activity as expected. And then we also did the N-terminal region mutations at a subset of the mutations from the South Africa. And they also have some reductions, but none of them, as expected, compare with the full spike. The full spike shows the most significant reduction.
Thank you.
If I could ask a question, actually, in your transcomplementation assay, using the cell culture and the mNeon as a readout,
right?
Are you capable of measuring protein production kinetics and or RNA production kinetics in those?
We haven't, yeah. We haven't really looked at specifically into this, but clearly, if you follow the mNeon signal, there is, that from very low and then the increase over the time, and of course the spread of the infections we can monitor. But specifically to your question, we haven't specifically looked at that, but we did northern blot on some of those just to make sure, the subgenomics and everything is behaving the way it is.
I guess the reason that I'm asking is I'm curious whether there might exist mutations that are not in spike, but in some other protein that are allowing the virus to replicate more rapidly, independent of what spike is doing. And do you know of any such mutants?
Yeah, so, when we perform the 10 rounds of passaging in the system to enrich or enhance its efficiency. We do see some mutations outside the spike. You know, the 3 mutations, only one of them is located in the spike.
And are any of those particular mutants coming up in nature, in human populations that you know of?
Yeah, we haven't looked at that, and even, you know, the mechanism and why those the other non-spike mutations occurred. We haven't really got time to look into that, and those are something is interesting to look at.